very much again for this kind introduction. I am very pleased to give this talk as a keynote speech at the conference. We are working, as you see, with transmission electron microscopes and immunolabeling in Graz. But today I will also show you a collaboration between the Agricultural Institute Center of Agricultural Research in Martonvasar, Hungary, and the University of Graz Institute of Biology in Austria. And I'll try to switch to the next slide. Yes. I'm pretty sure that you know that wheat is one of the 3 most important crop plants worldwide. And wheat is especially prone to global change. So as of now, we are facing 15% of the wheat production areas worldwide suffering from severe water scarcity or drought stress. And it is already alarmingly. However, by the end of this century, we might end up with up to 60% of wheat production areas suffering from this severe water scarcity worldwide. And we are interested in getting to know more about wheat drought tolerance, how to select for drought tolerant varieties. And we are also interested in what happens if the wheat production areas shift into higher altitudes or into other latitudes, and how that affects wheat growth, because their light composition might be different. So that is why we are interested in combining glutathione metabolism in plants and light, and combine that with stress response. So redox changes in plants as well as in animals and humans are a problem at some stage. However, they can also be beneficial, as we know for the past few years in plants. Reactive oxygen species, nitrogen species, and sulfur species may be also acting as signaling compounds to modify physiological processes. And just to give you a short introduction into how this works out in plants, I have chosen the very well-known in the plant science system Asada, Halliwell, and Foyer cycle of ascorbate and glutathione detoxification of hydrogen peroxide. I hope you can see the pointer. I will just quickly go through that cycle. So during drought stress, especially, but also during other oxidative stresses in plants, there is a pileup of hydrogen peroxide in the tissue, which originates from overstrained electron transfer chains from the photosystem too, but also from the respiratory chain in mitochondria, but especially from photosynthesis, which continues to work, although the stomata of the plants are closed due to drought stress. So the plant doesn't want to lose water through transpiration, but still photosynthesis is running. So the plant shifts to a process called photorespiration. And in the course of that, hydrogen peroxide starts to accumulate. And up to a certain extent, plants can neatly detoxify this hydrogen peroxide by transferring electrons from NADPH via proteinaceous and non-proteinaceous compounds in this cycle onto hydrogen peroxide to detoxify it to water, basically. So just to show you the subcellular compartmentation of plants for all of whom are not so familiar with transmission electron micrographs and the plant cell in general, I've prepared this slide. And what you see here is an ultra-thin section of a plant. It's 80 nanometers wide, and the picture was taken by my colleague Barbara Kofler. So first of all, we need, of course, the cytosol in a plant cell where all the organelles are in sort of floating. Then for plants, very important, of course, are the chloroplasts for photosynthesis. Also important is the vacuole. In the vacuole, soluble compounds can be stored, but hydrogen peroxide can also be detoxified, for instance, by ascorbate. In peroxisomes, this is the organelle that's very important also for detoxification of hydrogen peroxide, as we will see in a minute. And mitochondria, of course, are very important for cellular respiratory action and for the generation of ATP. And of course, plant cells also have a nucleus where the DNA is concentrated, and also DNAs then can be damaged by reactive oxygen species. So antioxidants such as glutathione are very important to protect DNA from oxidative stress, but also to protect membranes around the organelles, but also the cellular membrane that's close to the cell wall. From lipid peroxidation, because if lipids are peroxidized, the membranes may become leaky, and then cell compartments mix up in the cell, and that's not good, because in a cell, in a vital cell, we also have different pH in different organelles. So as I told you, hydrogen peroxide accumulates during drought stress, and we have shown that in Arabidopsis by cerium chloride labeling. This is the control section. There is no labeling visible, so there is no stress. However, if the plants are not watered for 10 days, you see small dark areas forming along the cytosol and also in the vacuole, especially along the tonoplast membrane that is the membrane that it surrounds the vacuole. So this is a way how to measure oxidative stress at the microscopic level in a transmission electron microscope. And my colleagues Bernd Zechmann and Maria Müller started to do these labeling experiments already 15 years ago. And several years ago, he and his student Elmin Heinecke and Nora Lushin Ebenkreuz, they looked into highlight stress in Arabidopsis and how that affects the subcellular compartmentation and the processes in compartments. And they have drawn this neat scheme for us. So reactive oxygen species started to pile up by highlight stress, first in the chloroplasts, because the electron transfer chain along the thylakoid membranes was overstrained. So plants had to shift to photorespiration, and then hydrogen peroxide piled up in chloroplasts, in peroxisomes, and later leaked out into the cytosol and eventually into the vacuole. And you see that in all these compartments, we have possibilities for ROS detoxification by catalase as well as by the antioxidants ascorbate and glutathione. What they also found was an increase in the activity of catalase, but with the highest intensity of light, I have to say. They also found tissue damage appearing, and it got more severe with more light intensity. And in the two mutant lines they also were examining, they even found browning of leaves and leaf senescence with the highest light intensities. Not surprisingly, light stress led also to this H2O2 accumulation, and which was made visible again by cerium chloride in Arabidopsis columbia, which is the wild type we use, again in cytosol, like here and here, and along the tonoplast in the vacuole. But you have to keep in mind that we are talking about a 10-fold intensity increase in light, which is very severe. If we look at ultrastructural changes in the different Arabidopsis types they were looking at, 14 days of high light treatment with this 1,500 micromole photon flux density per square meter and second of white light, they found that plastoglobally formed in greater numbers in the chloroplasts. And you can also see, compared to the control, that the membrane system within the chloroplast, the thylakoid membranes and the grana stacks start to somehow melt let's say they did not melt, but they were just reorganized. So the plants were trying to protect themselves and their chloroplasts by reducing the amount of thylakoids in the chloroplasts. If we now look into the two mutants, the PAD2-1 mutant is a mutant which cannot accumulate as much glutathione as Columbia, especially in chloroplasts other than mitochondria. So we see that there is even more plastoglobally formation compared to normal white light. And with Arabidopsis VTC21 mutant, which is an ascorbate deficient mutant, which can only produce up to 30% of Columbia ascorbate in the different compartments, you see that this is the most severe formation of plastoglobally. In our most recent project, together with our Hungarian cooperation partners, we thought it would be interesting also to include these three types of Arabidopsis together with our main plant of interest, which is wheat. And in the labeling experiments, we used the Chinese spring, which is a landrace that's cold tolerant. I quickly go over the light regimes with you now. Our partners in Hungary have controlled environmental chambers with LED lighting, which can be changed in the intensity, as you can see here, but also at the very fine scale of the light spectrum. So this is wheat. It's much older than the wheat seedlings we used in our experiments, but our cooperation partners were also interested in how the change in light intensity or the shift of light spectrum affects the quality of wheat flour and even bread dough. So that's why they also included blue light and more pink light. And just shortly, because I have to keep time in mind, I can tell you that with the shift to more blue light regime, the plants stayed longer at the juvenile phase, which is something that's already known. But also the bread dough quality and flour quality was different because there was a difference in the protein compounds. So gluten and glutenines were different under blue light conditions as compared to the white light conditions. And also the starch content was a little different. So for our experiments, we used for wheat low, normal and highlight and far red light treatment. And for Arabidopsis, normal, high and far red light treatment. Back to what we are doing in Graz for several years now to the subcellular localization of glutathione. We need to cut small pieces from leaves. This work was done in Hungary, and also the fixation step in paraformaldehyde, glutadialdehyde, and embedding in aloe white was performed in Hungary. After this polymerization step, the samples were transferred to Graz, and we took ultra-thin sections of the embedded plant samples. And what we do is we use a primary antibody which recognizes glutathione. It's a polyclonal antibody. And after incubation, we wash off excess antibody, which works essentially similar to ELISA technique. And then we incubate with a secondary antibody, which recognizes the primary rabbit antibody. So it's a goat anti-rabbit antibody. And on top of that, it's conjugated to gold particles. And the gold particles are needed so we can visualize the glutathione with the transmission electron microscope. To enhance the contrast of our images, we use uranyl acetate staining. And we take the pictures with a 4K camera, which is mounted on a Zeiss Libra 120 plus transmission electron microscope. To calculate how many particles we have per square micrometer in a certain compartment, we use Olympus Cell Sense Dimension imaging software. And now I'll show you how we do that. We trace a compartment such as the nucleus manually with the software tool. And then because the gold particles are electron dense, which means that electrons cannot pass through them in the microscope, they appear as very small black dots. And the software can calculate by the difference in grayscale between the black dots, respectively the gold particles bound to glutathione, and the rest of the compartment. So if we look at the results, we see that we do not find any under the increased light of 500 micromoles photon flux density, I have to say, which is only double the amount to our standard conditions. We did not find any changes in the system of thylakoid membranes. I hope you can see that here. They are still there. There was no major pileup of plastoglobuli due to that treatment. So we think that there was not a major oxidative stress occurring due to this short light treatment of 3 days on Arabidopsis. What we found, though, I have to say, is that there was an increase in mitochondria glutathione concentration and a decrease in the glutathione concentration in chloroplasts and in the cytosol. If we switch now to the far red light treatment, again, we found a decrease in glutathione in chloroplasts. I hope you can even see it from this image. There are less gold particles than in the chloroplasts here and here found, and also a decrease in the cytosol glutathione concentration. However, the mitochondria remained the same with respect to their glutathione levels. Again, far red light treatment did not cause any major formation of plastoglobuli or any major changes in thylakoid membranes. We have done a similar or almost the same experiment for wheat plants. However, the treatment was 10 days and we included a low light treatment as mentioned before. Again, as you see, the cell compartments look vital. There is no damage and also no major plastoglobuli formation or any damages to the membrane systems. This is a nucleus of wheat, which is much larger compared to Arabidopsis. And you see the hetero and euchromatin here. Just to give you a quick overview over the results, we found, as I mentioned, a moderate increase with increasing light intensity and far red light treatments in wheat compared to the normal and low light, but it wasn't as severe as in other compartments like the peroxisomes or nuclei and the cytosol, because as you see here, glutathione concentration is already quite high in mitochondria compared to other compartments. Our results also were reflected at the tissue level. So our cooperation partners measured concentrations of reduced glutathione, which is GSH, and oxidized glutathione levels, GSSG, or glutathione disulfide, in leaves, and also the ratio of glutathione disulfide to glutathione in the reduced form. So again, we see an increase in reduced and more severe increase in oxidized glutathione forms at tissue level in leaves, in highlight treatment, and also in far red light treatment. However, as you see, we are still at the highest point, around 0.15 in our ratio of oxidized to reduced glutathione, where we can assume that there is no major oxidative stress occurring in the plants. Our colleagues were also interested in the expression of several genes in Arabidopsis and wheat, and I just show you quickly the results for wheat. So the rate limiting step in glutathione biosynthesis carried out with the help of the gamma-glutamyl cysteine synthetase. And there we found surprisingly rather a decrease in expression with increasing light intensity or a shift to far red light. For the expression of the next step in biosynthesis of glutathione for the chloroplast associated gene and also for the cytosol associated gene, we found a trend, an upwards trend with increasing light intensity, no change for far red light treatment. And the only significant change we found in context to glutathione metabolism was an increase in the expression of glutathione reductase. And maybe you remember, but you will also see within the next slides, again, this enzyme and where it's at work in the glutathione cycle. So just to go also over the results for Arabidopsis, I can say that we found with the 3 days treatment a similar result compared to the 10 days treatment in wheat, for a rather similar result for PAD2-1. Maybe that's due to the fact that it's not able to produce that much glutathione and it starts to get into a situation where it wants to still have enough glutathione, and maybe also ascorbate was piling up, which we did not analyze in these experiments, but we know that PAD2-1 compensates the lack of glutathione with increased ascorbate levels normally under stress. But this is maybe the reason here. For VTC2 mutant and Columbia, we see that at the tissue level, there is basically no difference. Everything was increased, irrespective of reduced or oxidized form and also the ratio. And this is only partly reflected by far red light treatment, where we see only in wheat an increase in all these forms and in the ratio. But in PAD2-1, we also find an increased level of glutathione in the reduced form for far red light treatment. And for Columbia, which is the wild type and which is able to counteract stress the best among the three, we see that possibly these 3 days treatment is just a glimpse into an adaptation process towards the shift in light intensity or in the shift in light spectrum. So it's just an intermediate glimpse in the into the acclimation process. Our colleagues were also interested in looking into different wheat varieties with different drought susceptibility. So they took a drought tolerant variety called Plainsman and compared it to a drought susceptible one called Capelle de Pré. And just to show you one result, the electron transfer rate in the two was round about the same. So it's no major bit difference, but you can see that there is a little improvement in the recovery ETR rate in Plainsman under highlight conditions. So there is a trend that highlight intensities of 500 micromoles photon flux per square meter in second might enhance the recovery rate of this wheat variety. And in Capelle de Pré, this is a little less, but it's not a significant difference. However, what was intriguing was that the cysteine levels differed very much of the two varieties, irrespective more or less in Plainsman from the light treatment, but being high from the start and not dropping too much only at the recovery state under highlight, but never reaching the low levels of the other Capelle de Pré. And in Capelle de Pré, the cysteine level was a little bit enhanced. I have to say this is only a trend with the increase in light intensity. A similar result was found for the reduced glutathione concentration. It also was quite high in Plainsman, and it was comparatively low in Capelle de Pré. So to sum this up for you, we have an increased recovery rate for the drought-tolerant plainsmen and also recovery growth rate. This might at least partly be due to increased levels of cysteine and glutathione to counteract oxidative stress by drought. And I'd also like to sum up our labeling experiments for you. You already know this scheme. We found that glutathione was increased in the same compartments as was found from our colleagues several years ago. But the difference is that we do not see any major changes at the ultrastructural level. And there is no major difference between highlight and far red light treatment in wheat. If we look again at this detoxification model or pathway for hydrogen peroxide, highlight treatment with this double intensity that we used increased not only the two forms of glutathione, but also the expression of glutathione reductase. And in other experiments, my colleagues in Hungary also found an increase in cysteine, as I mentioned previously, but also an increase in gamma-glutamyl cysteine expression, synthetase expression, and glutathione synthase expression. So the whole glutathione biosynthesis and regeneration or reduction process, I have to say, for reduced glutathione is enhanced by highlight. We see the same enhancement under far red light. However, we could not find a transcriptional regulation in far red light treatment. Although cysteine levels also were increased due to the far red light. So there is some kind of boost from both light treatments. To conclude, I'd like to say that we were using an intensity of light that did not harm our plants. We could not see any damage at the tissue level or at the ultrastructural level, or any major changes in the thylakoid membrane system. Excess white light and low red to far red light might therefore be useful to induce glutathione metabolism, also other oxidants, antioxidants possibly. As we have seen previously from our colleagues, the anthocyanins are also increasing with increasing light intensity. And the drought recovery in the tolerant plainsmen might at least be due partly to this increase in glutathione and also the high levels of cysteine and glutathione present compared to Capelle-Depris. And with that, I'd like to draw your attention, if you're interested in further reading, to our other publications within this project, of course, also to past work that I have shown on the individual slides. I'm happy to answer any questions, but before that, I'd like to acknowledge the audience for listening. Again, the chair and the association for inviting me to give this keynote speech. And I'm very grateful, first of all, to Maria Müller, because she took me into her lab as a postdoc 3 years ago. And I learned a lot about ultrastructure, about electron microscopy and imaging and labeling from her. I'm also very grateful to Bernd Zechmann. He is a university professor at Baylor University in the US now, but he and Maria and him started this work on immunolabeling in Graz, and without him, we would not be where we are today. I'd also sincerely like to thank Stefan Möstl and Professor Günther Zellnig, because they keep our electron microscope running and are always very helpful with, when we need anything. And I'd like to also thank my colleagues mentioned here, Karina, Nadine, and Richard, who helped a lot with the sectioning and labeling of the samples and with image analysis. And of course, I'd also like to sincerely thank Gábor Kósi, the PI of this project in Hungary, also his students, Esther and Ákos, for doing the expression analysis for HPLC analysis, of course, the whole team in Hungary for maintaining the plants and growing them for us. And last but not least, I'd like to thank the funding agencies. I'm already running out of time, NKFIH in Hungary, and FWF in Austria. And thanks again for your kind attention. I'm happy to answer any questions.